them. And this can help you out in, streamlining what you have to answer, how you have to answer, and what exactly you have to study. Okay. So some probable questions from this initial chapter. One is, what is a cell? Draw a neat label diagram of the cell and its organelles. What is a mitochondria? What is its function? Then what are peroxisomes? What are the functions of peroxisomes? The Golgi complex, endoplasmic reticulum, what are different types of endoplasmic reticulum and its function, cytoskeletal structures, molecular motors, what is lysozyme, and then the nucleus and its function. Now, beyond this, we also have other questions like apoptosis and your transport functions, which are also there, which are part of the general physiology. Okay. So again, as I was mentioning earlier, starting June 5th, for the next 30 days, we have the exam mastery live sessions. So this is for you to, go through a quick revision, and so that you understand and get acquainted to all the important questions that are probably gonna come for your exams. Okay. So again, these are the various subscription plans which are available. You can use my code Dr. Shakthi 20 for 20% offer on subscription. So the main features of Auricle app, one are the live sessions, like the ones you're attending currently right now. Apart from that, there is a detailed recorded sessions of all the theory and the practical sessions as well. On top of that, there's a revision module also. And then there are notes, which are comprehensive for all the subjects, and MCQs of various degree of difficulty for your ease. Okay. And those of you who feel like you still need some sort of additional help, there are counseling sessions which you can book online in the Auricle website in order to get that personalized touch. Okay. So let's go ahead with general physiology. So I'm just gonna basically be using this PDF file which has been provided from the company, and for you also it will be made available, I'm sure, so that you can easily go through it, leaf through it, and understand what you need to learn. Okay, so we'll start off with some basic concepts. Okay, what is cell and all that. So again, for the exam level, we don't have to really know all of these. Okay, then question of what is a cell. Okay, so basically that is some basic question, like +2, +1 level sort of question. I'm sure most of you can answer it. You have a cell membrane, a nucleus, okay, nucleolus, and beyond that we have organelles. So at an MBBS level, how are you supposed to answer this question? Okay, we'll come to that. Let us draw a neat label diagram of cell and its organelles. So here you can see when you're drawing the diagram, there are few things that you have to keep in mind. One, we have to draw the nucleus. Okay. Once you draw the nucleus, you have to make sure that you draw a nucleolus and the chromatin structures which are there inside it. And beyond it, lamina should be there. Nuclear lamina should be there, right, covering it. Okay. And beyond that, again, you have pores that are over here. These are called nuclear pore complexes. So we have the nucleolus, chromatin, and then you draw the nuclear lamina. And beyond that, we have the nuclear pore complex. And beyond it, we have the endoplasmic reticulum. So over here, you can see this is the endoplasmic reticulum part which is there. That endoplasmic reticulum by itself are two types. You can see here, the ones which have granules in it, those granules are basically your ribosomes, and those are the rough endoplasmic reticulum. And near the cytosol layer, we have a smoother variant, which is there, that is smooth endoplasmic reticulum. So you have to draw the rough endoplasmic reticulum with the ribosome and the smooth endoplasmic reticulum as well. Okay, and beyond this, usually it is after this, you have the Golgi apparatus with first the cis and towards the end the trans. So from cis to trans, you have the Golgi apparatus. And from this Golgi apparatus, you have secretory vesicles which are budding off. So you'll have to draw these secretory vesicles coming out of it. Okay. And then we have your mitochondria structure. You have to draw it as a double membrane and outer membrane, inner membrane thrown into folds. Okay. Now, apart from these major structures, we have a lot of other structures which are also there. One is your cytoskeletal structure. So one by one, each of this, we're going to cover it, okay, in detail. So right now, at the outset, I'm just introducing basically all of these things. We have your cytoskeletal structures, including your microtubules, actin, myosin, intermediate filaments. All of these cytoskeletal structures are there. We have on the end centriole structure, which is important while your cell division is happening. Now, apart from this, we have two other structures like your lysosome and peroxisome. Okay, so these are various structures I want you to draw when this question of your cell is coming in. Okay. So again, can you see this? We have the rough endoplasmic reticulum. Okay. We have the smooth endoplasmic reticulum. We have the centriolar structures over here. And in and around it, you can see the microtubular structures. We have the mitochondria. We have the nucleus. Can you see the nuclear lamina over here that I was talking about earlier? And the nucleolus and the nuclear pore complex. We have the Golgi apparatus and structures like lysosomes. Also, we have peroxisomes. Okay. And your mitochondria. And the ribosomes also, you should draw some of the ribosomes on this part. Okay, your endoplasmic reticulum, the rough endoplasmic reticulum, but you should also draw free cytosolic ribosomes as well. Okay, and again, when you draw the cell, give it a polarity. So make this the basolateral segment, and this is the apical segment where you can give something of epithelial structures like villi or anything like that. You can just draw. So that will make a complete picture of your cell. Okay. Now, next question, what is mitochondria and what are the functions? So mitochondria, under that you have to talk that it is the powerhouse of the cell. Okay. It has a bilayered membrane, which is there, an outer membrane and an inner membrane. Okay. So a bilayered membrane, outer membrane, as well as a inner membrane. And the inner membrane is folded into projections called cristae. Okay, so here they have various enzymes which are responsible. So this is what you learn by chemistry. You have to incorporate that over here. Responsible for energy. So when you talk about functions of mitochondria, we can talk about your production of energy. Okay, so under production of energy, you're basically both these are actually one single point. Okay, so I want you to understand the structure like this. Okay, and we have these F1 particles over here. So that is what gives you the energy. Ultimately, we have the chemi-osmotic model based on which your F1 particle is there, through which H plus is moving to either side like this. And because of that, there is formation of ATP. Okay, so that is one function of your mitochondria, ATP. Apart from that, it is also seen that your mitochondria by itself can act as some sort of a calcium reservoir. Okay, now even in apoptosis, a damage that occurs in the mitochondria, which can be detected through a particular channel protein called MPTP. So that can also be helpful through the mitochondria. Okay. On top of that, you have to understand mitochondria is a circular self-replicating maternal DNA, which is present in the mitochondria. Okay. So these are all various points that you can write about when a question of mitochondria is coming. Okay, you can draw the diagram and then explain. So it's the same things. Okay, since this is a ATP, these both are same points. You talk about the chemiosmotic model and talk about apoptosis, and you talk about calcium reservoir and talk about the nature of its DNA. Okay, next structure is peroxisome. So this is often asked, and students don't have enough points to write about this usually. Okay, so when you take a look at peroxisome, I'll just show you a diagram of a peroxisome. So this structure that you see here, peroxisomes are usually something like this. Usually they call something like microbodies. So they have a membrane outside, a lipid bilayer, okay? And because of the function of your peroxisome, right in the center, you usually tend to have urate oxidase crystalline core. Okay, plasma membrane is there, and right in the center, urate core is there. So this is basically a structure. So the structure of lysosome also is going to be very similar to this. It has a bilayer, and inside it, instead of the urate crystals that you see, instead of that, you have all the hydrolytic enzymes which are present inside, which can get degranulated outside when needed. So that is your difference between a peroxisome and your lysosome. Okay. Another thing is peroxisome buds off from your endoplasmic reticulum. Lysosome generally bud off from your Golgi apparatus. Okay. So what are peroxisome again? They are membrane vesicles. Okay, just like lysosomes. They are pinching off from your endoplasmic reticulum. They contain various oxidative enzymes. Okay, oxidative enzymes which require oxygen. Okay, for example, catalase, urate oxidase. So many oxidase are present. So what are the functions of peroxisome? So the thing about peroxisome is, before talking about all of these things, the first thing I want you to understand is uh, peroxisome is one structure which is creating a controlled environment for peroxide formation and its degradation. So anything to do with, so inside the cell, you can't, produce peroxide, break it down. It'll affect all the cells, all the structures inside the cell. So you have to create a closed membrane structure, and inside which you can actually limit all of these oxidative enzymes can be released. Okay. And these hydrogen peroxides can be utilized and destroyed. Okay. And it's a controlled environment where your the peroxides which are formed can actually be utilized also for certain aspects. Okay, so that is it about your peroxisome main function. Peroxide in a controlled environment and its degradation breakage. Okay. Beyond that, it is also helpful in fatty acid synthesis and its breakage. So in beta oxidation, in one of the steps, it is helpful actually. Okay, so because of that also, it has a role in formation of bile acids. One intermediary step to a mature form, that change, you have your peroxisome being involved. Okay, so in some way, in lipid metabolism, it is involved. Lipid metabolism, it is involved. Okay, controlled environment for peroxides, free radical, all of that, and your formation of bile acids. Now, apart from that, they also helps in detoxification and your degradation of purine to uric acid. Okay, so one thing that you'll have to write is draw the diagram, explain what it is, explain how it is a microbody, and explain what are the substances that are generally there inside, and then you talk about what are the functions. You talk about one, control environment for hydrogen peroxide formation and its destruction. Then detoxification of substances, free radicals, for example, conversion of purine to uric acid. Then lipid metabolism, then bile acid formation. Okay. Now, apart from that, it what is written here, participate in the formation of myelin. Okay, I'd actually not go with that exact word sentence. It is seen that, in your neurons, if you look at it, next to the myelin, the number of, especially near the node of Ranvier, the number of peroxisomes are very high. Okay, so it helps in stabilization of axons, you could say more accurately. Okay, the axonal membrane stabilization and your myelin formation to support that your peroxides are playing a role over there. Okay, because whatever metabolic processes are occurring over there, for that, this plays a role. Okay, so what you have to understand is myelin. When you talk about myelin, the protein to lipid ratio, if you look at it, there's so much more lipid than protein. So it makes sense that peroxisome is important for myelin. Okay, so these are all different points that you can write under peroxisome. So I explained a little more, took a little more time for this because this is one question that comes and students do not have enough points to write under this. This is, correcting question papers, answer papers, and all this is generally what I felt. Okay. Then again, some studies do show that, in some way, it has enzymes which help in gluconeogenesis, especially from the fat component. So these are various functions of peroxisome. Okay. Now we have your Golgi apparatus and your endoplasmic reticulum. So I'll just, uh, it's easier to talk about it together. Okay, I just go to endoplasmic reticulum and then go to Golgi. That makes more sense. So endoplasmic reticulum, whatever transcription is happening from your nucleus, that will go, and now transcription, then translation should happen. At that point, the protein is formed. The protein that is formed is usually in the pre-pro form. That pre-pro form should be cut to give a pro form. And that pro form should be cut again to give the final protein. And that protein, based on whether it is a constitutive pathway or a regulated pathway, can directly go to the membrane or whichever else region it has to go, or it waits for a signal to result in regulated exocytosis. Okay, so endoplasmic reticulum is that point where after transcription, translation should happen. Okay, so the rough endoplasmic reticulum is important. Now, the ribosomes especially are important. It can happen either in the cytosol or in your rough endoplasmic reticulum. Okay, so whatever it is, the protein that is finally formed from your endoplasmic reticulum, okay, rough endoplasmic reticulum, usually should have a particular signal peptide. Only then it enters into your cell. Okay, so can you see over here? The mRNA is there, ribosomes are there. As it comes down, there is a particular signal peptide which is there. So when that signal peptide is there, then it detects it and goes inside the rough endoplasmic reticulum. If a signal peptide is not there, it does not go into the rough endoplasmic reticulum. Instead, it goes into the cytosol. So the protein complexes that come are of two varieties. Either it can go into the rough endoplasmic reticulum or it can go cytosol. Okay, the ones that go into the cytosol generally go either to the nucleus, mitochondria, peroxisome, like that. Okay, so the cytosolic ribosomes generally give proteins that go to your mitochondria, your nucleus, peroxisomes, and all that. On the other hand, the ones that go into your rough endoplasmic reticulum, for that some signal should be there, like the signal sequence, okay, peptide which is there, which has to be cut eventually. Okay, now as it comes, so why I'm explaining this, this helps cover part of a question of what are the functions of endoplasmic reticulum. Okay, now as it comes, it binds over here. Now it detects it, so it can go inside. Okay, but the translation and your translocation into this are happening at the same time. What is called co-translational translocation. So at the same time, can you see here the transcription, after that translation is happening. While the translation is happening, it goes into your endoplasmic reticulum at the same time. Okay, and at some point your signal peptide is cut, and finally your pre-pro protein is formed. Mostly pre-pro. Sometimes directly protein can also be there, but mostly a pre-pro form should be formed. Now from here, as you see, as it keeps growing, a lot of changes happen to it, which is one of the most important functions of endoplasmic reticulum. That is post-translational modifications. Okay, so we have post-translational modifications which are happening inside this. Okay, so what are these post-translational modifications which happen inside your endoplasmic reticulum? I'm so sorry. I think I was not showing the image. So this was actually the image. Can you see? This was the ribosomes. Okay. And these ribosomes, when it comes, this is the signal peptide which is there. And as it keeps coming like this, it goes inside. Okay, so that is your translocation into your endoplasmic reticulum. And this is your co-translational. While translation is happening, translocation is happening. Okay, once it is inside it, you can see a lot of changes that are happening to it, which is called post-translational modification. Now, the question is, what are these post-translational modifications that happen? One thing that happens is glycosylation. So over here, can you see how carbohydrate moieties are added? So that is your glycosylation. That is one thing. Apart from that, whatever protein is coming, you give disulfide formations. So intra disulfide formations are produced. Apart from this, hydroxylations can also happen. Something called GPI linkages. Okay, can you see this over here? So this protein structure which is there, you cleave it and attach it instead to a glycosyl phosphatidyl inositol. This is a GPI linked protein. Okay, all of these things happen inside your endoplasmic reticulum. And after that, we have your tertiary structure. Okay, protein folding takes place. And for that, you have your tertiary structure, which is there. Okay, so finally, tertiary and then quaternary, and you have the complete protein. So all of these changes happen inside your endoplasmic reticulum. So that is why when you talk about the functions of endoplasmic reticulum, you can talk about glycosylation, disulfide formation, hydroxylation, GPI linkage, tertiary structure formation, identification of the protein based on the signal sequence to differentiate it so that your protein transport can be adjusted accordingly. Okay, either it should go into the cytosol or inside the endoplasmic reticulum. Okay, but again, we still have functions with the endoplasmic reticulum. What happens is now, beyond this, say this tertiary structure, while it is happening, it gets misfolded or is unassembled. Some problems occurred. Can that come out of the endoplasmic reticulum? No. Again, that is a function here. So here, what happens is generally a structure called chaperones come bind to it, okay, and try to modify it and fold it properly. Okay, but it still doesn't work out. Then it attaches a protein called ubiquitin and goes for proteasomal degradation. Okay, so what does endoplasmic reticulum do? Not only does it do all of these things, but if it is misfolded also, it calls in chaperones, and with the help of chaperones, it tries to correct it. Even if beyond this it's still not folded properly, then ubiquitin comes in, tags it for proteasomal degradation. Okay. Now, from here, this question I've seen in MCQ, that's why I'm telling you this. From your endoplasmic reticulum, things has to go into your Golgi. So for that, we have cotamer proteins which are there, which are called COP. Okay, so from your endoplasmic reticulum to your Golgi, a COP2 is there for trafficking in that cotamer protein. Okay, and from your Golgi back to your endoplasmic reticulum, that is through COP1. Okay, so COP2 can take it to the Golgi apparatus from the endoplasmic reticulum, and from the Golgi back to the endoplasmic reticulum, that is COP1. Okay, and finally it's reached Golgi apparatus. What is the function of Golgi apparatus? So Golgi apparatus again has a cis side and a trans side. Okay, it has a cis side and a trans side. Cis side is closer towards the endoplasmic reticulum. Trans side is more towards the cytosolic gate. Okay, and you can see the budding vesicles which are coming. Okay, and finally it gets filled and it can go to the surface based on the type of signaling it is. Okay, so here what happens is, can you see here how different types of carbohydrate proteins, carbohydrate structures are added into it? So in the Golgi apparatus, more complex glycosylation beyond what you saw in your endoplasmic reticulum. Okay, transferring any form of sugar. Okay, and even N-glycan processing. So all of these things happen in your Golgi apparatus. Second function, again, your proteolytic, like cutting off some part of the protein. So we saw a pre-pro form. Pre-pro was cut in your endoplasmic reticulum. That reaches the Golgi apparatus. The pro, which is there, will be cut and the final protein is formed. So that cleavage of the pro protein is done by your Golgi apparatus. Okay, and after that also, now we have to pack and saw the vesicles based on whether it's a constitutive pathway or your regulated exocytosis. Okay, so such sorting out, okay, for protein transport and packing into the vesicles is also the function of Golgi apparatus. Okay, even neurotransmitter, whatever it is, any sort of protein which is there, or even a receptor, membrane protein. Okay, all of this, any ion channel. Okay, even like something like GLUT4 receptor, all of these are produced just like this. Okay, so that is the function of Golgi apparatus. So under Golgi apparatus, again, you can draw the diagram of Golgi apparatus with the vesicles budding off, the cis side, trans side. Okay, then you talk about the functions of your Golgi apparatus. Okay, so coming back to this endoplasmic reticulum, there are two types: rough endoplasmic reticulum, smooth endoplasmic reticulum. Okay, now the ribosomes are there in rough, agranular in smooth. So for protein synthesis, rough endoplasmic reticulum is very important. On the other hand, your smooth endoplasmic reticulum is more important for synthesis of your lipid substances. Okay, and you saw here Golgi apparatus. Again, packing department we already seen. Okay, secretory granules is basically the same thing. Uh, Again, site of formation of lysosome. So just like every vesicles bud off, one of the vesicles that bud off is your lysosome. Okay, whatever hydrolytic enzymes were synthesized, that comes out with a part of your membrane and buds off as a vesicle, forming lysosome. Okay, and again, complex glycosylation that we were talking about. So these are various functions of your Golgi apparatus and your endoplasmic reticulum. So you can see here nucleus, nuclear complex, endoplasmic reticulum, which is rough in nature because of ribosome, and this side you can see a smooth endoplasmic reticulum. Okay, not that accurate an image because usually the endoplasmic reticulum, the rough endoplasmic reticulum is closer to the nucleus. The smooth endoplasmic reticulum tends to be farther from the nucleus and closer to the cell membrane actually. Okay, just remember that when you draw stuff like this. Okay, so you talked about rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus. peroxisomes. Okay, now we'll continue with the remaining. So what is a cytoskeleton? So cytoskeletons are basically either a thin filament, a thick filament, okay, something like a microtubule or intermediate filament. So this is based on the size of the substance. Okay. So microtubules, as I was telling you, it can be a thin filament or a thick filament. When we say thick filament, we're talking about myosin. Okay, so myosin again has a head region, a hinged region, a tail is there. Now these myosins come together to form this myosin 2. There are different types of myosin, up till myosin 5, even beyond. Okay, so myosin can either exist like this or like this, and finally from that forms your muscle fiber. Okay, different parts of the muscle fiber. So that is one thing that is there. Structurally, myosin can help in contraction and all that. Then we have actin. So actin from a G actin, usually they keep coming together to form a F actin molecule like this. Okay, and one end it keeps growing, at the other end it keeps breaking down. So this actin also in many parts of movements that are there inside, like even a simple cell, if you take a look at it, it's pseudopod formation. Okay, that involves actin. If you look at the villi structure, they also have actins. Okay, so such subtle movements which are there inside that is dependent on actin. While contractile elements, whenever it is coming, your myosin is involved. Okay, change in shape and all that, that is your actin. Okay, so just think about it like that. So thin filaments are there, and we have thick filaments. And even for contraction, generally, this actin and myosin has to act together. And then we have intermediate filaments. These are between the thick and thin in terms of size. So there are substances like vimentin. Okay, so can you see this cell? So this is the nucleus part. Okay, so this was actually a rat cell nucleus, and if you look at it, you can see how much of fibrillar strands are there inside. These actually are your intermediate filaments. Okay, so they also help in maintaining shape, movement, and all that transport of substances. Okay, major transport is through microtubules. But apart from that, this also helps maintain the shape and all that. These substances are also there in your extracellular matrix as well, like fibronectin, laminin, things like that, which are there outside also. Okay, so these are your intermediate filaments. So when you talk about cytoskeleton, we have your thick filament, we have the thin filament, and we have the intermediate filament. Beyond this, we have your microtubules. So I want you to look at this one. Okay, what you see over here, not the ones on top, this one that you see here, this is a microtubule. So there are long strands of microtubule all over in your cells. Okay, so these are, just imagine that these are like your road work. Okay, how things are transported. It needs some pathway for it to be transported through. Okay, so microtubules are structures which actually help in transport of substances throughout your cell. One thing. Okay, they also make up part of appendages like cilia, flagella, and all. Okay, so substances like cilia, flagella, all those appendages, it forms part of that. It is also involved in cargo, transport of substances like between the cell. Okay, and to a degree, maintenance of cell shape and structure. Okay, and these are made up by smaller, as we saw earlier in the diagram, smaller structures called tubulin, which are there. Okay, so we had seen here, can you see this is the tubulin, alpha tubulin, beta tubulin? They are made up of tubulins. Okay, now we have intermediate filaments, which I already told you. So intermediate filaments not only are they important in maintaining your shape and all, but they also form part of cell anchoring mechanisms. Help anchor organelles together within a cell and also outside. And then we have actin and myosin, thin filament and thick filaments. Now, this is one question. What are molecular motors? Okay, so this is not exactly what I want you to write when a question of molecular motors comes. It's more to molecular motors than just this, what is given over here. Okay, so when you talk about molecular motors, you can talk about certain substances like this. These are called molecular motors. So we have one thing called the dynein, and then we have another thing called kinesin, another thing called myosin B. So on top of that roadwork we were talking about, which was microtubule, you need something, a lorry, to carry all these things. That is the molecular motor. Okay, if you want to carry it retrograde behind, okay, then you need dynein. For anterograde, you can use your myosin V and kinesin. Okay, so myosin, phi, and kinesin. So just for you to understand, these are the structures of these. This is how a myosin phi will look like. This is a kinesin, and this is a dynein. Okay, so I just want you to have a look at this. So can you see how it is moving? This is actually a kinesin which is actually moving with a cargo on top of your microtubular structure. Okay. Now, next question, what is the lysosome? Lysosome, again, we had talked about it, just like a peroxisome. It has a membrane structure, lipid bilayer membrane structure, but it has hydrolytic enzymes inside it. Okay, and what will that help in? These proteases will hydrolyze proteins. Lipases breaks down lipids to fatty acids and glycerides. Amylase, which is a saccharides, okay, sugar, carbohydrates to glucose. Nucleases are also, they hydrolyze nucleic acid into mononucleotides. So it helps in digestion of various substances. What does it help in digest? Either a cell or a cell which is there inside your body, or any extracellular components, like for example, bacteria coming. Okay, so it can degrade macromolecules, any worn out molecules which are there. Okay, and any excess proteins which are there secreted, that can also be removed. So under lysosomes, again, you can explain all this and maybe even draw a diagram. Okay, just like I had shown you peroxisomes. Now a nucleus. Okay, so when you talk about a nucleus, what is the structure of a nucleus? So this is a nucleus. Can you see a nucleolus over here? Okay. So nucleolus is one area where the transcription of the ribosomal RNA takes place. Then beyond that, we have a chromatin structure. So this chromatin structure, if you had to look at it, this is your DNA. That DNA, along with a histone octamer, forms a nucleosome. These nucleosomes condense together, packed together, form the chromatin. Now that chromatin gets condensed further, it forms a chromosome. Okay, so that is your genetic material which is there. It depends on what phase of your division cycle it is there. Euchromatin, heterochromatin structures are there. Okay, so you have the chromatin network which is there. And when division is needed, it becomes a chromosome-like structure in metaphase. Okay, and beyond that, we have the nuclear lamina. What is the importance of nuclear lamina? Again, during cell division, this nuclear lamina is very important because that goes off and helps form this nucleus again, reassembly after mitosis. Okay, and here also these pores are pretty important that we have particular pore complex mechanisms. This is the structure of a pore complex. We have a transporter subunit over here, right in the center. And on either side, there is a ring subunit. There's a basket-like structure over here. Okay. And there are thick filaments over here. You know what thick filaments are now? Okay. So this is what you call a nuclear pore complex. And this is important because whatever has to come into the nucleus requires certain nuclear localization sequence. So more specificity is there. Okay. And only certain things can come out of the nucleus as well. Okay, so if you have to draw a diagram of nucleus in detail, you can draw a diagram of nucleus with nuclear pore complex and explain all of this. Okay, and you can talk about the functions. So over here, what is mentioned is, very basic things that you learn in 10th standard, 11th standard. I want you as MBBS students to write a little more. Okay, so it forms the genetic makeup, synthesis of RNA, transcription. Okay, it forms the area where hormones with receptors, nuclear receptors can come act to produce signal transduction. Okay, it has hereditary information, contain mineral, control cell division. Okay, and I was telling you about nucleolus and how helps in the formation of ribosomes. And the parts of the nuclear lamina help reassemble it. And we talked about nuclear pore complex and how specificity for substances to come in and out is also there. Okay, and ultimately it controls major activities of the cell, protein production, and all that. Okay, so you can draw the structure of your nucleus and explain its function. Now, very rarely you might get a question like this. What is the more in biochemistry than in physiology, but sometimes it does come. So in that case, what is the cause of genetic disorder? How are they classified? So anyway, genetic disorders either can happen because of a mutation or some sort of variation that happen. Complete genome changes or specific DNA sequence is changed. Okay, so this is one type of classification you can do. This is a single gene disorder or a whole chromosomal disorder, or it can be a mitochondrial DNA. We already talked about how different the mitochondrial DNA is. Mitochondrial DNA disorders are there, or a multifactorial genetic disorder. It's still genetic, but a multifactorial. So it can be a single gene, a whole chromosome, okay, or a mitochondrial DNA, or a multifactorial genetic disorder. So I just wanted to give you some examples for this. Okay, if a question like this comes. So anyway, when you talk about a single genetic disorder, it can be autosomal recessive, it can be autosomal dominant, it can be X-linked. Okay, so under all of this, there are various examples. I'm just giving you a few examples, that's all. Okay, just one in each is more than enough. Then we have chromosomal disorders, like Downs, trisomy, and all that. Okay. Then multifactorial disorders are there. Now, apart from this, if you still want a little more to write as an answer, we again have the autosomal dominant, recessive, X-linked. Okay. All of that is your single mutations. Okay, and these are examples of that. Apart from that, chromosomal problems, one thing is non-disjunction, which can result in your Down syndrome. Okay, so those kind of problems can be there. Or you can talk about duplication, deletion, inversion with regard to changes, variations that happen. Okay, so you can classify genetic disorders into these various types. Okay, so I just take one small part, that is intercellular connections. So when you talk about intercellular connections, we have your gap junctions, your tight junctions, adhering junctions, desmosomes, hemidesmosomes, and we have focal adhesion. Different things are there. So this often comes as a question. So I'll try to show you one diagram. Okay, so can you take a look at this? When you look at this on the cell, first thing I want you to notice is this structure over here. So you can just imagine it as like, a Velcro by which you're attaching two things. One cell is there, another cell is there. You're tightly sealing them together. So that is your tight junctions. It determines how much tightness is there between two cells. If it's very tight, then paracellular flow is limited. Okay, so paracellular flow is determined by the tight junction. So what is the function of tight junction? Determines how much of paracellular flow is there. Okay, now how is the structure formed? It has structures like claudin, occludin, junctional adhesion molecules. Okay, so structures like claudin, occludin, junctional adhesion molecules are there. Also, two cells are there. On both the cell, we have claudin, occludin, junctional adhesion molecule, and they both are present like this, and they link to each other. Okay, and beyond this, there is another protein, now occludin, for example, and then it attaches itself to actin inside and actin inside. So this is some sort of connection where a transmembrane structure like your claudin, occludin, or junctional adhesion molecule is there, and an intracellular cytoskeletal structure like actin is there. So what does tight junction connect? Actin to claudin, occludin, or junctional adhesion molecule. And what does it do? It regulates the tightness, the paracellular flow between your cells. Okay. Now, apart from this, we have gap junctions, which are called communicating. Why are they called communicating? Because over here, the structure that is there is what is called connexon. This connexon is made of connexin. Each of those subunits is what is called a connexin, and these connexins together form a connexon, and they're there on either side so that ionic flow takes place between the cells. So easy movement of ions is happening between the cells, and hence they're communicating with each other with the help of ions. So that is gap junctions. Okay, so we done tight junction, we done gap junction. Here connexons, here claudin occludin junctional adhesion molecules. Now we have other things. Okay, we have adhering junctions, we have desmosome, focal adhesion, hemidesmosomes. What is the difference between them? So in adhering junctions and your desmosomes, just like we talked about claudin occludin junctional adhesion molecule, instead of that, there is one structure called cadherin. Cadherin. So whether it is a desmosome or a adhering junction, we have a cadherin which is there, which is connecting both of them. Okay. Now, beyond that, when we go inside, just like this, in ethering junction, we have actin. In desmosomes, we have intermediate filaments. That is the basic difference. Ethering junctions has a cadherin with actin. Okay, desmosomes have a cadherin with intermediate filaments. Tight junctions have claudine or claudine junction addition molecule with your actin. Just connexons, that is a gap junction. Okay, so what is the function of them? So, ethering junctions ether your cells together to form the epithelial monolayer. Otherwise, cells will grow very haphazardly here and there and can result in tumors, metastasis. So the thing is, the monolayer should be maintained. That is done by the ethering junction, ethers it. Okay. On top of that, your desmosomes help prevent against shear stress. When you go just push some cell like this, it just doesn't move away, rub off. That is because of your desmosomes. Okay, we have ethering junctions, we have desmosomes. How is it made? You can draw it also, these diagrams, and explain the function also. Now, extra edge, at least this I want you to know. As an extra edge, you can talk about focal adhesion and hemidesmosome. What are focal adhesion and hemidesmosome? These are basically just like ethering junction and cadherin, actually, with regard to structure. Okay, you just have to replace your cadherin with integrin. Cadherin with integrin. So, integrin with a intermediate filament connection, that is hemidesmosome. Okay, same way actin with an integrin, that forms focal adhesion. But the thing is, these are different. They are more with the outside matrix, not between cells. So these things that we talked about earlier are all connections between cells. Okay, these things, focal adhesions and hemidesmosomes, are not between the cells, but from a cell to the outside matrix. Such connections are focal adhesions and hemidesmosomes. So this is given nicely in your handout. Okay. So we have the cell junctions, occluding junctions like tight junction, communicating gap junction, and anchoring junction, in which we can talk about your ethering junction, your desmosome, hemidesmosome, focal adhesion. Okay, gap junction, we already talked about it. Function of gap junction, permitting your ions basically to pass through, any sort of ions. Okay. And this is also another important point. Apart from ionic exchange, because of that, a rapid spread of action potential. So in your cardiac muscle, okay, there's a whole syncytium. Atria acts as a separate syncytium, ventricle acts as a separate syncytium because of the presence of gap junctions. So when the atria is stimulated by an action potential, the whole thing starts depolarizing together very fast. Okay. Now, anchoring junctions, we have adherents, okay, and focal. These both have actin filament. One is cell to cell, the other one is cell to matrix. Then we have intermediate filaments. Intermediate filaments, desmosomes, hemidesmosomes. One is cell to cell, the other is cell to matrix. Okay, so can you see here between them with intermediate and cadherins, that is adherents and desmosomes. Okay, and here we got your focal adhesion where it is linked to actin and linked to your intermediate filament, that is hemidesmos. Okay. So these are the various topics which we were going to cover today. Okay. So now look through the questions again. What is a cell? Draw a label diagram of cell and its organelle. What is mitochondria and its function? What is the peroxisome? what are the functions of peroxisome, what is Golgi complex, what is the function of Golgi complex, endoplasmic reticulum, types of endoplasmic reticulum, and what is the function of that. What are the cytoskeletal structures? What are molecular motors? What is lysosome? And what is nucleus? Enumerate its function. And beyond that, the cellular communication, which is not given over here. Okay, so there you have it. Whatever questions gonna come from this topic, we covered it. All right. So thank you for watching. So if you wanna follow more such content, you can follow their Auricle's official handle, which is there in Telegram, Facebook, your Instagram, as well as YouTube channel. And you can download the app from the Play Store, which is readily available. Okay. So thank you. I think we can end the session.